Welcome to Chapter 25, Urinary System. General functions of the urinary system: elimination of organic waste, regulation of plasma concentration of ions, regulation of blood volume and blood pressure via the renin-angiotensin, roles and regulation of blood pH, and conservation of substances in the blood. The kidney is located in the retroperitoneal. It is in the superior lumbar region between T12 and L3. Gross anatomy of the kidney. Renal fascia is the outer layer of fibrous connective tissue that anchors the kidney in its place. Adipose capsule is made of adipose tissue and cushions and absorbs shock around the kidneys. Renal capsule isolates the kidney and prevents it from infection. Here is a drawing of a kidney. There are a few different structures inside the kidney. There's a hilus, which is an indentation in the kidney where the veins and nerves go through. There is an outer portion and a middle portion of the kidney. The outer portion is called the renal cortex. The middle portion is called the renal medulla, and it contains lobes. In the renal lobe, you will see renal pyramids, which contain renal papillae, which are pointy parts of the renal pyramid. Renal columns are in between the pyramids, and they're made of cortical material, which is the same thing that the cortex is made from. There is also a minor calyx and a major calyx. Both of these are tubes that carry the urine. The minor calyx is hooked to each of the individual pyramids. The major calyx is two or more of the minor calyx. There's also a renal pelvis, which is the funnel that collects all the urine and dumps it into the ureters. Blood supply. This is a rough idea of what it looks like when blood goes from the heart through the kidney and then back to the heart again. First, we start off with the aorta, then the renal artery, the segmental artery, interlobar artery, the arcuate artery, the cortical radiate artery. Then we get to the afferent arteriole, which then leads us to the glomerulus, then to the efferent arterioles, to the vasa recta, to the cortical radiate vein, the arcuate vein, the interlobar vein, the renal vein, and then through the IVC. These marked in the light blue, that are outlined in the light blue, these arteries and veins drain the kidney. The afferent arteriole through the glomerulus, then to the efferent arterioles and the vasa recta. These are associated with the nephron. Nerve supply. The renal plexus is a network of autonomic nerve fibers and ganglia. The renal plexus is supplied primarily by the sympathetic nervous system. The nephron is a functional unit of the kidney. A renal corpuscle is the bulb shape. Inside the renal corpuscle is the glomerulus with afferent arterioles, which carry blood into the glomerulus. And then there's also efferent arterioles, which carry blood out of the glomerulus. A glomerulus is a ball of capillaries. They are fenestrated and very leaky. Bowman's capsule is the outer portion of the glomerulus. It's the capsule shape. It is wrapped around the glomerulus and catches juice and sends down to the rest of the glomerulus. There is a parietal layer and a visceral layer. The parietal layer is made up of simple squamous epithelium. The visceral layer is made up of, it has podocytes, which wrap around the blood vessels like branches. At the end of those branches are pedicels, which are like toe-like structures that intertwine with podocytes. In between the openings of the pedicels are filtration slits. There's also a capsular space, which is located between the glomerulus and the visceral layer. This captures filtrate as it leaks from the capillaries. Here's a brief overview of how the filtrate flows down to the minor and major calyces. Proximal convoluted tubule goes to the nephron loop, which has an ascending and a descending limb. Also has a distal convoluted tubule, which leads you to the collecting duct. The next structure we'll talk about is the Juxtaglomerular Apparatus, which is also called the JG apparatus. This is a place where the ascending limb from the nephron loop comes in contact with the afferent arteriole. Components of the JG apparatus. It contains a macula densa, which is a patch of cells in the ascending loop, and those cells are chemoreceptors and osmoreceptors. These measure concentration of sodium in the filtrate, also measures how much dissolved solute is in the filtrate. JG cells act as mechanoreceptors, and they are blood pressure sensors for afferent arterioles. JG apparatus secretes renin and erythropoietin. Functions of the JG apparatus: it regulates the rate of filtration formation and regulates blood pressure. Types of nephrons. Cortical nephron is mostly in the cortex. A juxtamedullary nephron dips into the medulla and allows kidneys to produce concentrated urine. Capillary beds of the nephron include a glomerulus, which produces filtrate. Inside the glomerulus, you have afferent arterioles, which take blood in to the glomerulus, a larger diameter, and keeps the blood pressure up. Efferent arterioles, which brings blood out of the glomerulus and has a smaller diameter. Peritubular capillaries are responsible for reabsorbing the good stuff, for example, sodium and glucose. Vasorecta wraps around the nephron loop, helps extract more water, and makes urine more concentrated. In this section, we're going to talk about renal physiology, a few different sections. We'll talk about general information, glomerular filtration, tubular reabsorption, tubular secretion, regions of tubules and their functions, and also renal clearance. General information. About 1 to 1.2 liters of blood flow through the glomeruli per minute. That means about 125 milliliters of plasma is forced into the renal tubules per minute. Initial filtrate is blood plasma without protein. Urine is metabolic waste and unreclaimed stuff, such as nutrients, electrolytes, and most water is reclaimed. About 1.5 liters of urine is produced per day. Glomerular filtration. Hydrostatic pressure pushes filtrate through the glomerulus, equaling filtration. The filtration membrane squeezes fluid past. Capillary endothelium is made of fenestrated capillaries, which keeps blood cells in the vessels. Lamina densa is the basement membrane, which filters out the large proteins. Podocytes of the glomerular epithelium cause pedicels with the slit pores to catch proteins the lamina densa did not catch. Net filtration pressure. Glomerular hydrostatic pressure equals glomerular blood pressure. Forces opposing glomerular filtration is glomerular osmotic pressure, which brings filtrate back in, capsular hydrostatic pressure, which is smaller, and filtrate also gets brought back in. Glomerular filtration rate, GFR, is the total amount of filtrate produced per minute by the kidney as a whole. Factors affecting filtration rate: surface area available for filtration, filtration membrane permeability, and net filtration pressure. Tubular reabsorption is filtered every 45 minutes. It reabsorbs most of the fluids. Active tubular reabsorption is transported using ATP actively, such as glucose, amino acids, lactic acids, vitamins, and ions. These all will be absorbed in the nephron and should not be released through the urine. Processes of active tubular reabsorption are substances usually diffuses into tubular cells, which pump across the basal membrane of the cell. These are transported using the sodium-potassium pump. Then passively diffuses into capillaries. Passive tubular reabsorption is simple diffusion, no use of ATP, and it flows downhill. Also causes facilitated diffusion and osmosis. This also uses electrochemical and osmotic gradients. Osmotic gradients are set up by sodium 2. Water follows the sodium. Substances that are not reabsorbed. Reasons for this are lack of carriers, not lipid soluble, too large to pass through pores, substances such as uric acid and creatine. Tubular secretion functions: getting rid of substances, eliminating substances not in filtrate, for example, drugs, and other substances that are unwanted but have been reabsorbed passively, or excess potassium. Another function of tubular secretion is control pH of the blood. Substances secreted by tubular cells are such as hydrogen ion, potassium, creatine, and organic acids. Regions of the tubules and their functions. Proximal convoluted tubule is the first part of the tube after the glomerulus. Its function is the largest reabsorber. Most stuff is absorbed. Reabsorbing is active transport via ATP dependent carrier, which reabsorbs sodium, glucose, amino acids, and vitamins. By penocytosis is small proteins. By passive transport is chloride and bicarbonate ions. By diffusion is urea. By osmosis is water. Water moves. This is secreted into filtrate, which causes hydrogen ion as needed to keep blood pH correct. Loop of Henle, which is also known as the nephron loop, has a thin segment, which is the first part of the descending limb. It dips into the medulla. The thick segment is in the cortex and is the ascending limb. Functions of the nephron loop: it reabsorbs water, sodium, and chloride. The descending limb is freely permeable to water but not salt. Ascending limb is impermeable to water, permeable to sodium and chloride. Distal convoluted tubule and collecting duct have principal cells which targets targets aldosterone, causing sodium absorption. Functions is low permeability to water and sodium when hormones are not present. Selective reabsorption of sodium and hydrochloric acid. Hormonal controls of antidiuretic hormone. Little reabsorption of water unless ADH is present. When ADH is present, pores enlarge, water resorbed and turns into concentrated urine. When ADH is absent, pores close, which causes the collecting duct to be impermeable to water and dilutes the urine. Aldosterone is released from the adrenal cortex in response to decreased blood pressure or hyponatremia, which is low sodium levels, or hyperkalemia, which is high sodium levels. Antinatriuretic factor is the antagonist to aldosterone. ANF, which is atrial natriuretic factor, is released from the cardiac cells in response to high blood pressure or high blood volume. This inhibits sodium reabsorption and causes more sodium and water into the urine. Renal clearance. This is how fast the body clears stuff out. The volume of plasma from which a substance is completely cleared in any given time. This generally takes about 1 minute and is important for testing different types of drugs.